trade is endemic to southern western Ghats. Is distributed from Wayanad to Chikmagalur. It is commonly called as rose sapphire berry and locally it is called Malankuruvi. The limited literature exists on the morphology and phytogeography of Symplocos roseae, so it is necessary to validate its medicinal property. Various Symplocos species are scientifically recognized for their antioxidant, antimicrobial, antidiabetic, anti-inflammatory, and antivenomous property. Its evolutionary proximity to this established species, Symplocos roseae, is strongly hypothesized to possess similar pharmacological potential. According to APG system 4 of classification, the Symplocos roseae bed belongs to the family Symplocaceae under Ericales. The main objective of my study to evaluate the phytochemistry and therapeutic potential of Symplocos roseae. The main objectives of my study: the separation, isolation, and characterization of phytochemicals from different parts of plants. To evaluate the medicinal properties such as antioxidant, antivenomous, antimicrobial, antidiabetic, and anti-inflammatory property. To identify the bioactive compound with therapeutic uses by in silico method. And coming to the methodology adopted to carry out my entire research work. Plant collection. This distribution map show where this plant is located in Kollam district. The plant samples of Symplocos roseae were collected from Rosemela, which is located in Shenduni Cross Division in Kollam, Kerala during October 2022. Identification of plant species were done with the help of Dr. Stephen. Then herbarium preparation. Herbarium was prepared as per standard method and voucher specimen has been submitted to the herbarium of Sree Narayana College, Kollam. Morphological analysis is large shrub or small tree up to 7 meters in tall. The leaves are simple, alternate, margins are serrate and glabrous. Flowers are racemose in fluorescence and pink in color. The fruit is narrowly oval, seed is grooved. Soft-solid extraction. The fresh leaves and stems of Symplocos roseae were collected, dried under the shade and ground into fine powder. The powder was extracted using soft-solid apparatus with NHC, ethyl acetate and methanol based on its polarity. The extracts were filtered and concentrated using rotary evaporator and stored at 4 degrees Celsius. Then several qualitative and quantitative phytochemical analysis were carried out using standard protocol. The qualitative phytochemical screening were used to detect the presence of secondary metabolites like alkaloids, glycoloids, flavonoids, tannins, phenolic compound, terpenoid, coumarins, and saponins. The quantitative phytochemical analysis were used to detect quantify the total phenol, flavonoid, terpenoid, and alkaloid content. An overview of the pharmacological activity of this study is shown here. The extract of Symplocos roseae were investigated for antioxidant activity, antimicrobial activity, antibiofilm activity, anti-inflammatory activity, antidiabetic and antivenomous activity to assess its therapeutic potential. The antioxidant assay, DPPH radical scavenging assay, hydroxyl radical scavenging assay, and FRAP assays were used to analyze the antioxidant activity of plant extract. Then antimicrobial activity. Agar well division method is used to analyze the antibacterial activity of methanolic extract of Symplocos roseae stem and leaves. 8 bacterial strains including both gram-positive and gram-negative were tested. Pseudomonas aeruginosa, Escherichia coli, Serratia marcesinis, Staphylococcus aureus, Bacillus subtilis, Proteus vulgaris, Salmonella type, and Proteus mirabilis were used. Streptomycin is served as a positive control and DMSO is used as a negative control. Then antifungal activity. Slightly modified dual culture method is used to analyze the antifungal activity of plant extract. 3 fungal strains were tested using Aspergillus brasiliensis, Fusarium oxysporum, and Candidum albicans. Then determination of minimum inhibitory concentration. The Risazolin assay method is used to analyze the minimum inhibitory concentration of methanolic extract of Symplocos roseae stem and leaf against Pseudomonas aeruginosa. Then antibiofilm activity. The minimum biofilm inhibition concentration of methanolic extract of Symplocos roseae stem and leaf against Pseudomonas aeruginosa was determined using crystal violet assay. Then anti-diabetic activities analyzed using alpha amylase and glucosidase inhibition assay